Hello and welcome back to Chem 4 lecture. This week we're moving on into chapter 13, where we're going to cover our first set of biomolecules. So the way this is going to work is chapter 13 is on carbohydrates, which are commonly known as sugars, a pretty common type of biomolecule and a very important type of biomolecule. Now we're going to cover these because we covered aldehydes and ketones in the previous chapter. And that sets us up to understand the chemistry of carbohydrates, which is really interesting for a large variety of reasons. Then the next chapter, we go back to organic chemistry, and we're going to talk about carboxylic acids and esters, and I think amides as well, if I remember right. So yeah, so that just kind of sets it up for the next few chapters here. So we have our first biomolecule here. As you'll see, it's still a lot of carbon, hydrogen, oxygen. So not too different from what we've been looking at, but it has its own set of things and rules that we want to learn about. So, okay, so let's go ahead and start with carbohydrates. Now, you know carbohydrates in your diet as carbs. These are basically the main source of energy that we have in our diet. All of these carbohydrates are going to be made from carbon, hydrogen, and oxygen. And they can also be called sugars, commonly. And then the scientific name for that is saccharides. And we'll have a few different words for it here in a couple slides. So carbohydrates like glucose, for example, are these biomolecules that are synthesized in plants. Now, plants have a special set of enzymes that we're going to talk about in the future that basically take in sunlight energy and use that to break the bonds of carbon dioxide and water and turn those into these sugar molecules, these carbohydrates. So starting with glucose is kind of the simplest example, although there's a bunch of different ways that it can go from there. Now, the way that this works is basically you take 6 molecules of CO2 plus 6 molecules of water, and it takes a lot of energy, right? This is energy from sunlight. Photosynthesis then produces glucose, and glucose is going to be our first monosaccharide that we're about to talk about, or carbohydrate. Now, in that process as well, it produces oxygen. Now, interestingly, we do basically the reverse process. Animals and other non-plant sources basically take the carbohydrates that are, from plants essentially, and we break them down back into carbon dioxide and water. So the sunlight, the plants take in the sunlight to build these complex molecules. Then animals like us eat the complex molecules, break it back down into CO2 and water. And so there's a sort of carbon cycle there with that. Okay, so there's a few types of carbohydrates that we're going to talk about. First, we're going to talk about monosaccharides. That's where we have just one carbohydrate molecule, but carbohydrate molecules can link up, as we're about to talk about. So then the next type of important carbohydrate molecule is what we call a disaccharide. That's where we have 2 monosaccharides that are linked together. For example, sucrose is an example of a disaccharide, whereas glucose, galactose, fructose those are all monosaccharides. Again, disaccharides, that's where we link 2 of these carbohydrate molecules together. Now, it doesn't have to stop at 2, and in fact, it can go on much beyond that with what we call polysaccharides, which can contain up to thousands of these monosaccharides all linked together. So these are things like starches, cell walls are made out of this as well. There's a lot of different examples of these that we'll talk about in a little bit. Okay, but of course, we're going to start with the simplest, which is monosaccharides. So this right here, this is erythrose, which is a very simple monosaccharide. So we've seen monosaccharides a couple ways now. We're going to have this cyclical structure that we'll talk about more at the end of the chapter. And they also have this straight line structure that we're going to start explaining things this way because it's a little simpler. Now, there's 2 different types of monosaccharides. There's ones that have an aldehyde. That's, of course, where the carbonyl is at the end of a chain, and that carbon is in the carbonyl is bonded to a hydrogen. If they contain an aldehyde group, that monosaccharide is known as an aldose. So aldehyde becomes aldose. However, you can also have sugars that have a ketone group instead. So here, this ketone group is in the second carbon from the top. And this carbon in the carbonyl is bonded to 2 other carbons. That's why we know it's a ketone. And those are known as ketoses. So this is the fundamental breakdown here of these monosaccharides. It's either an aldose or a ketose. Now, these monosaccharides also have variable lengths of chain. So both of these now have 4 carbon chains here. This is erythrose, the aldose, and this is erythulose, the ketose. They're both 4 carbon chains. So that's just one example, though. Glucose, for example, is a 6 carbon chain. There's 5 carbon chains. 7 carbon chains, I think, happens rarely. I don't think we're going to talk about any of those, but the main ones that we will be talking about are 5 and 6 carbon chains for the monosaccharides. But no, it goes from 3 to 7. Now, also very commonly with saccharides, or monosaccharides, I should say, all the other carbons in the chain have hydroxyl group. Hydroxyl groups are really common in these organic molecules and in carbohydrates in particular. So we expect to see a lot of these hydroxyl groups around here. We'll talk about that in a minute. Okay, here. So again, most of the time, the types of monosaccharides that we'll talk about are going to be split into 2 categories. That's aldose, which has an aldehyde group, ketose, which has a ketone group. And then from there, we're going to classify it based on the number of carbon atoms that they have in the carbohydrate chain. For 3 carbon atom long chain, that's called a triose. For 4, that's a tetrose. 5 is pentose, and 6 is hexose. And then you combine those 2 words in a way that basically tells you, first off, do you have aldehyde or ketone, and then how many carbons are in the chain. So for example, aldopentose means that you have an aldehyde group, and that's a 5 carbon long monosaccharide. A ketohexose, on the other hand, would have a ketone, and it would be a 6 carbon long chain. So this is just a few different ones here. You don't have to memorize these necessarily, but just to kind of explain it here. So glyceraldehyde is an aldotriose. It's a very small carbon chain. So it's a very small monosaccharide. This is present as part of the metabolism of sugars that we have in our body that we'll talk about later. Threose is a aldotetrose. So it's again an aldehyde group with a 4 carbon chain. Ribose, which is important in terms of ribonucleic acid, which we'll again talk about in the future, has 1, 2, 3, 4, 5 carbons. So it's a pentose with an aldehyde group. So it's an aldopentose. And then finally, fructose, as I mentioned, another important monosaccharide, has 1, 2, 3, 4, 5, 6 carbons in its chain. So it's a hexose, and it's got a ketone. So it's a ketohexose. Okay, now don't just take it from me. Go ahead and give it a shot for the sample exercises. So make sure, take a look at the sample exercise packet or put this in your notes here and tell me what type of sugar is it. So we want to classify these monosaccharides in terms of aldose versus ketose, and also tetrose, pentose, hexose. So go ahead, pause the video here, give it a shot, and when you come back, we'll take a look at the answer. So go ahead, pause. Okay, here we go. Let's go ahead and take a look. So first up, for A, we have 1, 2, 3, 4, 5, 6 carbons in the chain. So it's a hexose, and it's an aldehyde group. So we would call it an aldohexose. Then for B, we have 1, 2, 3, 4, 5 carbons in the chain. So it's a pentose with a ketone. So that's a ketopentose. Okay, now we have to talk about a particular property of carbon molecules, or carbon atoms, I should say, here. And this is going to be a little confusing unless you have a molecule structure kit, which I realize I didn't bring out yet, but I will right now. If you have a molecule kit, that's great, and you should follow along with what I'm about to show you. But if you don't, that's okay too. What we're going to do is I'm going to build it and try to show it to you on camera as well. So hopefully that works out okay. But what we want to talk about now is chirality. So the basic fundamental principle here is that based on, okay, think about it this way. You have a left hand and you have a right? And they are analogous to one, right? But are they the same? Do they do the exact same thing? Well, not really, right? I mean, they don't have the exact same structure, right? If I try to overlay my left hand on top of my right hand, no matter how I do it, I'll never get it perfectly lined up. they are mirror images of one another, but they are not the same. And this can also happen with carbon atoms, as we're about to see. Okay, so real quick review. Before we talk about stereoisomers, let's talk about structural isomers that we mentioned in the previous chapter, or I think 2 chapters ago now. We know that molecules can have structural isomers. That's when they have the same molecular formula, but the bonding arrangements are different. So for example, ethanol and dimethyl ether are both C2H6O, so they have the same molecular formula. However, the bonding arrangements differ. In ethanol, you have an oxygen that's bonded to a carbon and that's bonded to a hydrogen, whereas in dimethyl ether, that same oxygen is bonded to 2 carbons. So clearly they're different structures, they have different bonding arrangements, they're gonna have different properties. Now, that can also happen, for example, with aldehydes and ketones. So if you take the formula C3H6O, that could represent propanal, here, the aldehyde, 3-carbon aldehyde, or it could be propanone, which is better known as acetone. Again, both of these have the same chemical formula or molecular formula, yet we can see that they would have different properties because we have an aldehyde and a ketone. This aldehyde could be oxidized to a carboxylic acid, whereas this ketone could never do that. Okay, so stereoisomers go one step further. They have the same molecular formula, and they're not structural isomers because the atoms are still bonded in the same sequence. The only thing that's different about the 2 stereoisomers is that the arrangement, the way that those atoms are arranged in space is different. So this is how we identify this, and we're gonna introduce a couple words here that we want to add into our vocabulary here in organic chemistry. When you take a mirror image of an organic molecule, and then you take that mirror image and you cannot match it directly over, just like with our hands, we can't match them directly over one another. We would say that those are non-superimposable. In other words, they can't be directly superimposed on top of one another. And then they are chiral. So we are looking for chiral carbons. And even though this is a lot to say, we have some examples that are going to back you up. We have the kits from lab that are going to help you with this. And we're going to have a very simple way of identifying these. So just hold on, keep watching, and we're going to make it through this. Okay, so chiral molecules have the same number of atoms, again, the same bonding arrangements, but those bonding arrangements are just differently put together in space. And the way that we can find this is we can find a non-superimposable mirror image. So again, non-superimposable means you can't put one directly over the other. So like, think about it this way. If I just take 2 carbon atoms that are not bonded to anything, these are directly superimposable, right? I can put one directly over the other and it perfectly matches it. With my hands, I can't do that, right? If I try to put them directly one over the other, instead, they never match up. It's only, they only become mirror images of one another. Okay, so some examples of chiral and achiral everyday objects. Gloves, just like your hands, of course, are chiral. A glass, which is perfectly round, is achiral. There's no problem. You can directly superimpose that glass over the other glass. It matches up exactly. You can fit it in, probably stack it in to make a bigger stack of glasses. And your shoes as well, having left and right feet, are achiral, or I'm sorry, they are chiral as well, because you can fit your left foot in the left shoe, the right foot in the right shoe, but you can't flip them. Okay, here we go. Now, molecules are chiral and therefore have stereoisomers when they have at least one or more chiral carbon atoms. Okay, now this is easy to identify because all we need to find a chiral carbon atom is we are looking for a carbon that is bonded to 4 different groups. So to identify if a carbon is chiral, you look at that carbon and ask, okay, what is it bonded to? It's bonded to, let's say it's bonded to a hydrogen. Okay, that's 1. It's then bonded to a hydroxyl group. That's 2. Then it's bonded to a methyl group. That's 3. And then it's bonded to an ethyl group. That's 4. That carbon, because it's bonded to 4 different groups, would be chiral. That's all we have to do to identify these chiral carbons. Okay, so now let's go ahead and build an example. So let's go ahead and build, this is now, I guess, bromochloroiodomethane. We're going to build it 2 separate ways. So let's go ahead and give it a shot. So we've got carbon bonded to hydrogen, and then hydrogen bonded to, let's use the green for chlorine, just like they do here. I'm going to try to match it up here. Now, this presents another challenge, which is that I am also in a mirror image here, stuck inside your computer. So things get a little wonky here, but I will do my best to represent it. We'll use blue for iodine like they are, and we'll use red. We don't have brown here. Oh, no, we do have brown. We use brown for bromine over here. Okay, so I've got now I have 4 different groups bonded to this carbon. I've got hydrogen here. This was iodine, chlorine, and bromine. So you'd agree with me, those are 4 different things bonded to the chiral carbon, right? Okay, 4 different things, great. Now what I'm gonna do is I'm gonna build the mirror image. So I take a new carbon center, okay? And I'm gonna put the hydrogen bond this way, which is the way that I have it here. Then the mirror image. So 1, 2, 3, 4. I'll just put all these single bonds in here first. Okay, so the blue goes up. And then green is back from there. And then brown is forward and front. Okay, great. So let me try to show you these here. Okay, there we go. So now we have 2 mirror images. Would you agree with me here? You see that the hydrogens are in the center, the chlorines are out facing towards you, the bromines are back facing towards me, and then the iodines are up. So it doesn't exactly match what we have in the screen, but it still gives you an idea. Now, if these were the same molecule, what I could do is I could arrange them such that they exactly superimpose over one another. So that's what I'm going to try to do now. So we see that we have these hydrogens here, right? So let's go ahead and try to put the hydrogen on top of the hydrogen. Okay, great. All right, so we got there. So you see the hydrogens on top of the hydrogen. Right? Okay, so the hydrogen is superimposed on the hydrogen. And then the iodine's also superimposed on the iodine. So we're close. We're already 2 out of the 4 groups there. Okay, but now the problem is this bromine is superimposed on this chlorine. So I guess I'll switch it, right? And then when I switch it, now I've got the chlorine on top of the chlorine. And back here, you can see the bromine's on top of the bromine. So now I'm done, right? Oh no, what happened? Oh my goodness. Now the hydrogen of this one is on the iodine of this one, and the iodine of this one's on the hydrogen of this one. Oh, that can't be good, right? Okay, so let's flip it again. And okay, great. Now the hydrogen's on hydrogen, iodine's on iodine. So it's done, right? Oh no, wait, the bromine and the chlorine are mashed up again. So no matter how many times I rotate this or move this around in my little computer screen here, I can never get them to superimpose on one another. That means because of the arrangements in space, these are different molecules. When we have these mirror images that can't be superimposed, we call these molecules enantiomers. So we have 2 separate enantiomers here that are stereoisomers of one another. Okay, now let's say that instead of iodine on both of these, we had a hydrogen. Or here, they're using bromine, so I'll use bromine as well. Well, no, I ran out of bromine, so let's go ahead and use hydrogens. So this is just to point out the fact that here, if we have now these molecules that have 2 of the same group on it, hydrogens like this, if we're ever in a situation where, okay, so the hydrogens are on top of the hydrogens, but now this chlorine is on top of this bromine, you flip it now, and now take a look at it. Oops, sorry. Bromine's on top of bromine, hydrogen's on hydrogen, hydrogen's on hydrogen, chlorine's on chlorine. So these are superimposable mirror images. Because these are superimposable, that means that they're achiral. And again, you don't have to like do this, you don't have to build a little structure each time you have this question. All you need to recognize is, okay, I have this carbon is bonded to a bromine. That's one type of thing. It's bonded to another bromine. That's the same thing. So it's just one thing, plus a hydrogen, plus a chlorine. That's only 3 different things. If your carbon's only bonded to 3 different things, it's achiral. It's not chiral. Okay, now I know that was a lot. If you need to, definitely pause, back up the video, and try to go through it again. And of course, if you have any questions, make sure to ask down in the comments below. And what I want you to do is identify these as chiral or achiral carbons. And again, we're just looking at the carbons that were centered on here. So just focus on those. And again, if you have one chiral carbon, it's a chiral compound. So try to identify A, B, and C as chiral or achiral. Pause the video now, and when you come back, we'll take a look at the answer. So go ahead, pause. Okay, we're back. So for A, we look at our central carbon. It's bonded to hydrogen, 1, chlorine, 2, methyl group, 3, ethyl group, 4. That's 4 different things. That's chiral. How about this one for B? This carbon is bonded to hydrogen, 1, hydrogen, 2, methyl, so that's 2, chlorine, that's 3. So it's only bonded to 3 different things. Therefore, it's an achiral carbon. And then finally, this carbon is bonded to a hydrogen, a chlorine, a bromine, and a methyl group. That's 4 different things. That's a chiral carbon. Okay, now we're going to talk about how we're going to draw these carbohydrates in 3 dimensions, or I'm sorry, in 2 dimensions, with keeping in mind that they are 3-dimensional molecules. So the way that we do this is there's a special type of notation for carbohydrate compounds that's called a Fischer projection. And again, it's a 2-dimensional representation of what we know is a 3-dimensional molecule. Now for this one, we're not going to have to draw the dashes and wedge structures each time because by convention, we would have a lot of dashes and wedges to draw. So we're going to show you how this works just with doing it on a flat piece of paper. Okay, so starting with basically what we've already seen for carbohydrates so far, those are Fischer projections. This is where we have the most highly oxidized carbon at the top. So that's the aldehyde or the ketone at the top. And then the vertical lines going down, the vertical lines, those actually represent basically dashes that should go into the page. Okay, and then the horizontal lines are wedges that should come out of the page. So let's go ahead and take a look at this. So for L-glyceraldehyde here, this is a simple aldotriose. We've got the aldehyde at the top, then we've got a carbon in the middle, and then a CH2OH at the bottom. Now, we just need to draw the aldehyde at the top. We don't have to worry about any chirality for that one because check it out, this carbon only has 1, 2, 3 bonded atoms. So that's trigonal planar. So we're not worried about any chirality with that. This carbon in the middle, however, we are going to worry about that chirality because here it's bonded to aldehyde, an alcohol, a hydrogen, and a hydroxyl group. That's 4 different things. That's a chiral carbon. So by convention, what we do is we take these that would be wedges sticking out, and we're just going to put them flat lines, or straight lines, I should say, on the horizontal here. So the horizontal lines, we know that those bonds should be coming out of the page towards us. Sorry, so going out of the page towards you. The vertical lines are the opposite. Those should be coming into the page towards me. And this is the Fischer projection here of this L-glyceraldehyde. Now, if you take the mirror image of it, because we have a chiral carbon, you know, here we have the hydrogen on the right and the hydroxyl group on the left. The mirror image then has the hydrogen on the left and the hydroxyl group on the right. So these are both glyceraldehyde. This one is L-glyceraldehyde, and this is D-glyceraldehyde, for reasons that we'll talk about in just a little bit. But because of these chiral carbons, these now are non-superimposable mirror images. Okay, yeah, so we're ready to talk about D and L notation. Okay, so D and L isomers are assigned according to the position of the OH group on the chiral carbon that's farthest from the carbonyl. So here we've got an erythros again. This was one of the ones that we talked about before. Erythros has 2 chiral centers. We've got a carbon here and a carbon here. This carbon has its hydroxyl group on the left side, so we call it L. This is the L isomer of erythros. Now, if you have that hydroxyl group on the right, on the other hand, we call that D-erythros. So this actually works out pretty well because L, of course, you can think of as left. So hydroxyl on the left is L for the farthest chiral carbon. If it's on the right, then it's D. And the way you can think about this is use dextro. Again, that's the Latin word for right. So if you think D means dextro, dextro is right. You know that the hydroxyl group should be on the right for the D isomer. Okay, so let's go ahead and try to figure out, are these mirror images here that cannot be superimposed? So do we have a set of enantiomers here, that is, they're mirror images that are non-superimposable, or do we have non-chiral compounds, or are they not related to one another? Now, I will give you a hint here. Both of these pairs are mirror images of one another. So you need to ask yourself, are they superimposable or not? So go ahead, pause the video here, take a look at it, and see if you can determine which is the set of enantiomers. So go ahead, pause. Okay, let's take a look. So the first thing that we want to figure out, if we have a pair of enantiomers, is do we have a chiral carbon? If we don't have a chiral carbon, then we don't have enantiomers. So looking at the central carbon here, we can see it's bonded to an alcohol up here, a hydrogen here, that's two different things, methyl here, and bromine here. That's four different things, so this is a chiral carbon. The other molecule here clearly represents a mirror image, where the bromine is still on the bottom, the alcohol group is still on the top, but now the methyl group, which was on the right, is now on the left. The hydrogen, which was on the right, on the left, is now on the right. So that is a mirror image of a chiral carbon. That is an enantiomer. So the way that you can think about this is just imagine drawing a mirror in the middle, okay? And what would happen is basically you get the substituents closest to each other are going to change, and the substituents farthest away from another swap as well. So the first swaps for the last, and then the second swaps for the third, is the way that you can think about it. So for both of these, you see that swap, that's a mirror image. And again, we have a chiral carbon, so it's enantiomers. All right, how about this for B? Well, we have a mirror image, right? So if we draw a mirror, we have a mirror image. So we're going to draw this in. This methyl group is on here. This hydrogen's over here. However, do we have chiral carbons? Are these carbons chiral? They're not, right? This carbon is bonded to two hydrogens. That means it's only bonded to it's only bonded to three total different groups. So if it's not bonded to four different things, it's not chiral. Okay, so yes and no. Okay, that's easy. Okay, next up, this hopefully is a little easier. Sample exercise 4. Identify the following Fischer projections as either L or D isomers. So again, what you're looking for is a chiral carbon farthest away from the aldehyde or ketone, and ask yourself, is the hydroxyl group on the left or is it on the right? Let's go ahead, pause the video, give it a shot, and we'll take a look at it. Okay, so for A, start up here, we've got this aldehyde here. We're looking for the farthest chiral carbon. So this is a chiral carbon, this is a chiral carbon, this is a chiral carbon, this is a chiral carbon. You can do this really easily because you can see, okay, the way you want to think about it is basically this. Okay, so for this carbon here, we know it's bonded to a hydrogen. We know it's bonded to a hydroxyl group. That's two different things. Then don't just look at the atom that it's bonded to. So you might say, oh, this is a carbon and this is a carbon, so those are the same. That's not the way it works. Here it's bonded to this CH2OH down here, and here it's bonded to C4H6 or 7 and O4 compound up here. So this is different from this, right? Would you agree with me on that? So that's why we would call this, the third different group, and this is the fourth different group. So this is a chiral carbon in that way. So now we look at the farthest chiral carbon, which we just identified, which is this one right here. The hydroxyl group is on the right of it. So because it's on the right, that means it is a D isomer. There we go. On the other hand, for B, now the OH group is on the left. So that is the L isomer. I guess it's a different saccharide here, but you get the idea, because this hydroxyl group is on the left, and this is the farthest chiral carbon from our aldehyde. You might ask yourself, well, what about this carbon here? Well, this carbon is bonded to two hydrogens, right? It says CH2. Since it's bonded to two hydrogens, that means that it's not, it can't be chiral. Okay, now, what does this all mean? or why does this even matter? Really? And the truth is, it matters a whole heck of a lot. Because for biological compounds tend to be chiral. In other words, they tend to have either a left-handed or a right-handed nature. So I mean, I don't know what to tell you. It just really matters. The enzymes that make up our body are chiral. The carbohydrates that we have in our system are chiral. And then the way that these bonding arrangements are arranged in space have to fit into a very small space. So we're going to try to build up our knowledge of biological chemistry towards the end of this class as we're getting there now. And this is kind of one of the first steps where we want to kind of explain something in the future where we are learning it right now. Basically, biological systems fold in a certain way based on the chirality of the molecules that make them up. Basically, like think of it as having our two chiral molecules here. If you have this molecule, like if you have a protein that fits the chlorine, bromine, and iodine in this exact way, like this, where the chlorine's on top, the iodine's sticking towards me, and the bromine sticking towards you. Well, if you took the enantiomer, it's never going to fit. The chlorine fits up here, but now all of a sudden the bromine and iodine don't fit down here. So, because of the nature of the complexity of our biological systems, these enantiomers matter a lot. And in fact, a lot of times, so yeah, just to give you an idea, again, if you think of 4 different groups here for this particular carbon, where you have triangle, sphere, cube. So just like, in preschool, we're trying to fit the shapes into the box. But here, it's a very complicated box that you're trying to fit it into, and those only fit with a certain arrangement. For the mirror image, it's no longer going to fit that way. If it doesn't fit with your enzyme, then it's not going to react with the enzyme. The whole process shuts down. So let's look at an example. Parkinson's disease uses a compound called L-DOPA, which is related to dopamine. It's converted to dopamine in the brain. If you use D-DOPA, on the other hand, so L versus D-DOPA here, that's not effective at all. In fact, your body doesn't recognize it, and it just kind of passes right through. And that's like the best you can hope for, is where one of them works and the other one just doesn't fit. A lot of times, drugs that are produced that are mixtures of enantiomers can actually have a poisonous effect. In fact, if you research thalidomide, we can talk about it a little more in class, but thalidomide was a chiral drug that was produced to prevent morning sickness. And in the original way that they made one pure enantiomer. When they got approved and started producing it industrially, they made a mixture of enantiomers, and the other enantiomer was actually a poison for children. And a lot of children were born with birth defects, and many women miscarried based on the poison that they were given as a drug. So again, that's the worst that can happen, basically, is where you have something that's a really useful drug on the one hand, and on the other hand, literally on the other enantiomer is a poison. Okay, so let's go ahead and take a look at Fischer projections of monosaccharides. Now, I want to say up front a really important thing here, which is that I'm not going to require you to memorize these structures. In fact, I will be giving you the D-glucose, D-galactose, and D-fructose structures on the test. However, you will need to be able to manipulate them. Now, another piece of good news is that for the next 2 tests, so the organic chemistry test and the biochemistry test, those will both be open note. So you will be able to take notes on these chapters and use them in your test. Okay, so again, we talked about Fischer projections. We use them a lot to represent carbohydrates. That's where we have the most oxidized group at the top. We can easily identify chiral carbons because we see what's bonded on the horizontal lines, what's bonded on the vertical lines, and we see an alcohol group at the bottom, the CH2OH group, and that's achiral. Oops, okay, wait, so didn't we just talk about this, the chiral carbon from the farthest group? Did I just double this up? Okay, all right. Now we're gonna look at very particular carbons, monosaccharides here. I think I accidentally put this in twice, but this is good because we can actually see real monosaccharides that we're going to use. So ribose is up first. We have L-ribose and D-ribose. L-ribose is where we have the 3 hydroxyl groups on the left side. And of course, what matters is basically that the last one is on the left. So that tells us that it's L-ribose. And the D-ribose is where that final chiral carbon has the hydroxyl group on the right, like what we're seeing here. Now, the relationship between L and D ribose is that they are enantiomers. So each of these chiral centers is flipped. Now, for glucose, we see now that it's a 1, 2, 3, 4, 5, 6 carbon chain. And L-glucose has the hydroxyl group on the left. D-glucose has the hydroxyl group on the right. Okay. So now go ahead and take a look at this. This, I'm telling you, is a sugar called mannose, which does come up every so often. It is relatively important, not as important as glucose or galactose, but mannose does show up every so often. And I want you to identify this as either the D or the L isomer of mannose. So go ahead, pause the video here and tell me, is this D or L? Okay, well, again, all you have to do is from the Fischer projection, you're going to go to the carbon, the chiral carbon that's the farthest away from the aldehyde in this case. So that's all the way down here. And then you look at the hydroxyl group, it's on the right. That means it's going to be a D isomer. So we would call this D mannose. Okay, next up, let's go ahead and identify. Now, you don't need to name this carbohydrate, but why don't you classify it according to the scheme that we talked about at the beginning? So this is like a pre thing here. Identify the number of chiral centers, and then identify the monosaccharide as either D or L as far as an isomer goes. So again, is it an aldose or a ketose? Is it, tetros, pentose, hexose, and the number of chiral centers, and then finally D or L. So go ahead, pause the video, and when you come back, take a look at the answer. Okay, let's go ahead and get started. So starting from, we see we have an aldehyde up here, so we know it's an aldose. And then we have 1, 2, 3, 4 carbons in a chain. So that is a tetrose. So we call it an aldotetrose. The number of chiral centers, well, this carbon is an aldehyde, so it can't be chiral. This carbon is bonded to 2 hydrogens. That can't be chiral either. This carbon is chiral because it's bonded to aldehyde this side, this mess down here, a hydrogen hydroxyl group. This carbon is chiral as well. Same reason, bonded to a hydroxyl group, a hydrogen, and a alcohol down here. So we have 1, 2 chiral centers, and then we look at the farthest chiral center from the aldehyde. We see that the OH group is on the left, so that should be the L isomer. So 2 chiral centers, L isomer. Okay, now let's talk about where we interact with glucose and fructose. And in particular, we're going to talk about the actual ones that we use. So the sugars that we use in our bodies and are used in the world are right-handed. So these are D isomers. So D-glucose comes from fruits, corn syrup, or honey. It's the aldohexose that we saw on the previous slide. It's also known as dextrose, or blood sugar can be called blood sugar as well when you measure it in your body. So if you measure, blood sugar levels, you're looking for glucose in the blood, basically. Now, glucose itself will build up into sucrose, which is a disaccharide, lactose, again, a disaccharide, maltose, a disaccharide, and then other polysaccharides like cellulose, which makes up cell walls, and glycogen, which is an energy storage matrix. Fructose comes from sucrose, and it is a ketohexose. Same formula here, C6H12O6, but a structural isomer of glucose. So the way that they're built is different. It is the sweetest on if you take a sweet scale of what's the sweetest carbohydrate, this is one that scores the highest. It scores twice as sweet as sucrose, which is table sugar. Yeah, and so you see it in honey. Both of these are present in honey, and that's what gives honey its sweet taste. Okay, now related to those is D-galactose. Galactose is half of the disaccharide lactose, and it has a similar structure to glucose. The only difference is that the OH on carbon 4, so 1, 2, 3, 4, is on the left side for D-galactose, whereas it's on the right side for D-glucose. So again, that doesn't seem like a big deal, but because of that, they have totally different properties, and they can be used in different ways. So if you have galactosemia, that means that an enzyme that usually converts galactose to glucose is missing. So for most of us, we process galactose by first turning it into glucose and then sending it through our metabolic pathways. However, it's possible to miss that enzyme that does that. And in that case, you would not be able to process galactose. And I believe you would actually have galactose as a toxin in that case. Okay, so now let's bring up hyperglycemia and hypoglycemia. So this is probably a condition that you'll see a lot if you're practicing health in the future, because this is related to one of the most common illnesses in the United States, which is issues with blood sugar regulation and in particular diabetes. So for a normal, for an average level of glucose in your blood, you normally have a range of about 70 to 90 milligrams per deciliter in your blood. And the glucose tolerance test measures blood glucose for several hours after ingesting glucose. So this is funny. This happens like if you get pregnant, they'll test you for your metabolism. You drink this whole thing. And what they do is they measure your blood several different hours, several different times following that ingestion. So for a normal or typical response, you see a bump of about an hour and then a return back to a normal level for the blood glucose level. If you have the condition of hyperglycemia, the blood glucose shoots way high up and then never recovers to the normal level. Hypoglycemia, basically you have a small bump and then return to very low levels. So diabetes mellitus can cause hyperglycemia, where the pancreas is unable to produce insulin. And because of that, insulin's not there, you don't process glucose as quickly as the normal patient does. So the glucose levels can rise to as high as 350 milligrams per deciliter. Note that that's about 5 times as high as it should be. Diabetes symptoms would include thirst and excessive urination. So basically your body's just trying to get rid of that excess glucose by drinking a lot of water and then eliminating the extra glucose through the urine. And then also an increased appetite. So you're eating more, but you're not processing the sugar as well. So you're losing weight. And sometimes diabetes can be the consequence of excessive weight gain. Hypoglycemia, on the other hand, is when the blood glucose level rises and then decreases rapidly to levels as low as 40 milligrams per deciliter. So about half as much as the typical patient. Again, this could also result from a related problem where you have an overproduction of insulin by the pancreas. And these are just symptoms, really. These are just, you're not necessarily finding the root cause. There can be a lot of root causes that lead to these. The symptoms that would appear would include dizziness, where your brain isn't getting enough energy, and so you don't have the processing power that you normally do. General weakness and muscle tremors. Again, your muscles are missing out on the sugar from the blood, so they're not as able to handle just everyday activity. And then oftentimes for hypoglycemia, you have a diet that's prescribed with several small meals that have high protein and low carbohydrate diets. Okay, all right, so sample exercise number 7. Now, sample exercise number 7, again, as I mentioned, I'm going to give you the structures for D-fructose on the test. So D-fructose, D-glucose, and D-galactose, and you'll also have them in your notes as well if you want to. So going back up here, you can use this structure of D-fructose. Go ahead and draw it in your notes right now. So pause the video now, draw the D-fructose in your notes, and then make it into L-fructose, okay? So remember, L-fructose is going to be the mirror image of D-fructose. So let's go ahead and go back here. You can rewind the video to get that structure, go back to sample exercise 7. So go ahead, give me now in your sample exercise packet, the Fischer projection for D-fructose and then for L-fructose. So pause the video, write those down, and we'll take a look at the answers when we get back. Okay. All right. So D-fructose, hopefully you just drew it down from our notes from earlier. It's, again, a ketohexose. And here the OH is on the left for the first chiral carbon, OH on the right, OH on the right for the second and third chiral carbons, followed by CH2OH. So only 3 chiral carbons here. L-fructose is the enantiomer of that. So you just take the mirror image. Again, just imagine drawing a mirror here between the two. The H on this one becomes the, on the right, becomes the H on the left on this one. OH on the right becomes OH on the left. OH on the right becomes OH on the left. And this is L-fructose, the mirror image of D-fructose. Okay. All right. Now we have one more type of structure that we want to talk about, which is what's called a Haworth structure. Because as it turns out, Fischer projections are really useful for comparing these carbohydrate monosaccharides. But as it turns out, when you actually have the molecules, they don't really behave in that way. Instead, what happens is they form a ring. So pentose and hexose sugars tend to form 5 or 6 atom rings. And that's what we're going to look at here for the Haworth structure. Okay, so first up, let's go ahead and learn how we're going to draw a Haworth structure. So starting from D-glucose. So D-glucose looks like this. Again, this can be in your notes. That's no problem. Now, what you do is you take D-glucose and you're going to turn it 90 degrees sideways. Okay, so take it clockwise, 90 degrees. And what's important is first start by numbering. The aldehyde carbon, or whatever carbon's at the top, is going to be carbon 1, and then number down from there. So 1, 2, 3, 4, 5, 6. It's important to do that because we need to keep track of these carbons as we make our chain. Now, the really important oxygens and carbons to note here are going to be here, this hydroxyl group that's on the farthest chiral carbon. And here, this, of course, aldehyde group here. This is what's actually going to change when we start actually drawing the Haworth projection. So take your Fischer projection, turn it 90 degrees clockwise. Okay, next up. Start by then basically turning it into a ring. So carbon 1 can stay there. Then carbon 2 comes forward towards you. Carbon 3 is level with carbon 2. And then carbon 4 goes back away from you again. Now carbon 5 goes back even further into the page, and then carbon 6 is back away from you. Now, here's a tricky one. This is really tricky to do. But what you're going to do next is, you see how you've got the OHs down here? And those are the OHs that were on the right side. Again, it was on the right side, now it's on the bottom. If it's on the left side, now it's on the top, because we rotated it clockwise. Or sorry, I guess we rotated it clockwise, like this to this. Okay, now what's going to happen is carbon 5 is going to rotate. Where we have the hydrogen on top here, we're going to move the hydroxyl group in line where carbon 6 was. So now carbon 6 is going to be pointing up, so that oxygen off of carbon 5 is in the chain. This is what it looks like after you rotate. So now, keeping track of our carbons, we've got 1, that's still the aldehyde. 2 coming in out of the page towards you. 3, again, level with 2. 4 going back into the page. Now it's in the plane with the page. 5 going back even further. And now this oxygen here, this is where we're going to take this oxygen. Oh, and this oxygen is going to form a new carbon-oxygen bond with the aldehyde. So this OH group now is going to form a new oxygen-carbon bond here. This that was an aldehyde is now going to be an alcohol. So this oxygen that was an aldehyde is going to be an alcohol. And that alcohol group can either point up or it can point down. If the alcohol group points up, that's what we call the beta isomer. If it points down, that's called alpha isomer. We'll talk about this more in a little bit, and we'll have some time to practice it too. Now here, if we have the alpha isomer can actually interconvert into the beta isomer. because this chain can open and this process can go on again and again. But basically, all of your glucose is either going to be the alpha cyclical form or the beta cyclical form. Very little in solution is going to be open chain, like the Fischer projection. Instead, it's going to be converting between the two forms if you just have a solution of glucose. Now, for glucose in particular, as it turns out, the beta structure is more stable than the alpha structure. So the beta structure makes up about 64% of glucose in solution. Okay, so when you put a solution of glucose, or when you put glucose in a solution, the cyclic structures have the chance to open and close. Now, they don't open and close often, but because they can, if they're in solution long enough, eventually they will. And so the alpha isomer will rotate and turn into the beta isomer, and vice versa. So this is basically an equilibrium process. And at the end of it, you end up with about 36% concentration of the alpha version of glucose and 64% of the beta version of glucose. Now, you don't have to memorize those. That's not like a particular, that's not general to all monosaccharides. That is very specific to alpha glucose and beta D glucose. Okay, now fructose is a little bit harder, but the way that it works is basically the same. Because fructose is a ketohexose, that means it's got a ketone. So its Haworth structure is actually going to be a 5-member ring rather than a 6-member ring. So it looks more like a cyclopentane, like this. Now, the way that this works is here our ketone is going to turn into an alcohol. And that alcohol, whether it's pointed down, would be for the alpha structure. And if it's pointed up, would be for the beta structure. So once again, the same thing is true here, where this is our generic template here for a Haworth structure. Carbon 2 is going to form, is going to be like at the corner of this pentagon here. Carbon 3 comes forward. Carbon 4 stays in the same plane as carbon 3. Then carbon 5 goes back towards the paper. And then carbon 5, that's our final chiral carbon. That's the one that's going to form the new oxygen-carbon bond with its oxygen from the hydroxyl group back to carbon 2. And that's what we actually see here. Now, again, carbon 2 can have its hydroxyl group pointed down for the alpha structure or pointed up for the beta structure. I'm not going to require you to do this on the test. But why don't you go ahead and give it a try with galactose. So use the same method that we used for glucose to draw the cyclic form of D-galactose. Give me both the alpha and the beta form. So go ahead, pause the video, look back at what we did with glucose. Check your notes and maybe rewatch the video a little bit to get an idea of what you want to do. Remember, we're going to walk it through as well, but I want you to give it a try first and see how you do. So go ahead and pause the video here. When you come back, we'll take a look at how we get the answer. Okay, let's go ahead and take a look. So first up, remember the first thing we're going to do is we're going to take our Fischer projection and rotate it clockwise by 90 degrees. So this is carbon 1, 2, 3, 4, 5, and 6. When we rotate it clockwise, we get carbon 1, 2, 3, 4, 5, 6. The things that were on the right are now on the bottom. The things that were on the left are now on the top. Okay, and now we're going to fold it forward. Remember, carbon 2 comes forward. Carbon 3 stays in the same plane. Carbon 4 goes back to the original plane. Carbon 5 goes back, and then we rotate it. Carbon 6 is pointed up now. And the hydroxyl group off of carbon 5 is now going to form an oxygen-carbon bond to our original aldehyde over here. And what ends up happening is if that hydroxyl group points up, that's the beta form of beta D-galactose. If it points down, that's the alpha form of alpha D-galactose. So these are the two different cyclic forms that can be drawn from from D-galactose. So go ahead, give this a little bit more practice. There's one of these on the homework as well. On the test, I will be giving you one of these problems, or I give you a Fischer projection, and I ask you to draw the Haworth projection. So make sure that you have this in your notes as well, since you'll be able to use those on the test. Okay, any questions that you have, make sure to ask them in the comments below, or when you see me, I know this is a complicated subject, but you can get started working on the homework now. And I think you'll do fine as long as you really make sure to maintain your notes from here on out this semester. Okay, until then, I'll see you soon and have a good day. Bye-bye.